Hello everybody, I'm Filippo Valle from the University of Turin in Italy, and this virtual poster is about multi-omics topic modeling for breast cancer classification. It's on the study of RNA sequencing data or gene expression transcriptomic data, and to study this kind of data we use topic modeling. Topic modeling is a framework of community detection, but it is more informative. For example, we can have not just clusters, but we have probability distribution that tell us which gene is important in which kind of tumor, for example, or what are the drivers of these cancer subtypes. We will see later examples. I will show you an example of topic modeling that uses networks. This approach is more powerful than other approaches, and the performance are better. These topics are more informative than clustering because you can use them to create, for example, cohorts of patients and use this to study, for example, the survival probability of patients. And with this work, we release a Python tool that you can use today to integrate multiple omics or study transcriptomics or any omics data, even integrated together. Let me explain how this kind of model works. We have gene expression data, and from this kind of data, we can build a bipartite network like the one shown in this slide. On one side, we have genes, on the other side, we have patients. We run our model, and the output are topics on one side. Topics are clusters or lists of genes with a probability distribution associated to them. So, for example, we can tell you which is the most important gene in a given topic. On the other side, we have clusters, so a group of patients with the same cancer subtype, for example, and we can associate to each sample, each patient, a probability distribution over topic. So I can tell you which is the most important topic in a given patient and how this topic is composed, which are the genes most important in this topic. Now let's have a look at the output of this kind of model. On the left, you can see the clusters, so the group of patients that the model puts together. Each column is a cluster, and each color is a breast cancer subtype annotated from the Cancer Genome Atlas. As you can see, there are clusters in which all the patients share the same breast cancer subtype, so at least clustering is well performed. If we look on the right, in this plot on the y-axis we can see a score that measures how well the subtypes are identified. On the x-axis is the resolution, so the number of clusters we are looking for. And the different lines are different kind of models. The blue ones, the one that reaches the higher score is our model. And as you can see, this kind of approach outperforms all the other models we have tested, like latent ratio allocation or hierarchical clustering or weighted gene correlation network analysis. One of the advantages of using network-based topic modeling is that if we start with the bipartite network we shown you before, we can simply append other omics, for example, like microRNAs data or copy number variation data or mutations data, simply adding a new branch to the model. So we no longer have a bipartite network, but we have a tripartite, pentapartite, etc. And we call these n-partite stochastic modeling, NSBM. In this slide, I show you the results of this integration of omics. On the left, I show you the clusters of patients with tumor or without tumor, and as you can see, they are well separated. This is a tripartite network with the patients, genes, and the microRNAs. On the right, it's a bipartite network with genes and patients, and as you can see, the tumor and the normal samples are not as well classified as the model which has the integration of the microRNAs. One of the advantages of using topic modeling instead of clustering is that we can have this probability distribution with the topic given sample, and we can use them to create cohorts of patients. If, for example, we can separate patients which have a topic upregulated or downregulated. And as you can see in this figure, if a patient has a topic upregulated, is our survival probability are smaller than patients which have the topic downregulated, which have a higher probability of surviving. Before going to conclusion, let me mention and thank my whole group in Turin. So let me conclude. I will show you that topic modeling is a very informative way to perform clustering. I've shown you that this network-based topic modeling can outperform other approaches of clustering and also other approaches of topic modeling like latent direction allocation. I've shown you that these probability distributions can be used to create cohorts of patients and study, for example, the survival probability of patients. And with this work, we release a tool we call NSBM. You can download it from Anaconda and use it today. If you have a multi-omics problem and you want to integrate multiple kind of data, multiple omics, you can use it right now. In the QR code shown above me, you can download our paper. Thank you for your attention. Bye-bye.